And I just couldn't put it down. I couldn't put this down any more than I could put down my interest in bio threats or starting the company. And so this was another epiphany. This was the moment. And again, just like walking on the campus and deciding I'm going to medical school, from one step to the other, I said, I don't know where this is going, but I'm gonna do something. In 1993, my wife and I stayed in Palau to just look around. We hired a guide. He took us to a rock island where there was something jutting out from the shore. It was a 65-foot wing. And that 65-foot wing essentially changed my life. The markings indicated that it belonged to an American World War II plane. I just felt a loss. Nobody knew what happened to the crew. Somewhere there was more to that plane and more to the story. If nobody knows anything about what happened, I was going to find out.
Welcome to Titans of Transition, featuring real-life stories, anecdotes, and inspiration about pivotal career moments from leaders who have been there. Join host Joe Miller, former scientist and senior technology leader turned executive coach, as he unpacks transitional moments to help inspire change.
Welcome to the Titans of Transition podcast. I'm really pleased to have Pat Scannon with me today. Dr. Pat Scannon is truly a Renaissance man, if I may say so myself. He really has focused in three domains in his life and his career. One is the development of innovative therapies for diseases with large unmet needs. Another is in the application of biodefense policy, incorporating these technological areas in response against intentional, natural, and accidental threats to the American people. And finally, in searching for MIAs, missing in actions, and POWs from World War II, as the founder of the BentProp project, now Project Recover. Pat, welcome to the podcast today.
Good to be here.
Pat, the focus of Titans of Transition is just that, looking at those who have been very successful in managing transitions in their career and in their life, and specifically to encourage our listeners to navigate those transitions, both to sense them coming, to respond to potential transitions that either are thrust upon them or they feel the need to make, and learn from other notable leaders how to deftly, shall I say, make those shifts. So in doing that, let's explore your background and career. And I'd like to start off by talking about the area of biotech and XOMA.
Okay, so I had received my PhD in chemistry from UC Berkeley and realized almost intuitively that there was a growing interest in the area of biology and chemistry, this interface. And there really weren't a lot of advanced degrees or programs. Molecular biology, when I graduated in 1972, was a very nascent kind of science. And recombinant biology hadn't been discovered yet. So I figured naively or otherwise that going to medical school would be my entry into biology. And so I did that. Being a Georgia boy, I went back to the Medical College of Georgia and worked 12 months a year and was able to finish my medical degree in 3 years. And then did my internship and residency in the United States Army, in the Army Medical Corps, at the Letterman Army Medical Center. Did my residency in internal medicine, and then the Army decided that with my background in chemistry, I should go and do medical research, and I was assigned to the Letterman Army Institute of Research, where I was working on in trying to develop hemoglobin as an artificial blood substitute. Well, about that.
Pat, I just interrupt you a second. Just if we could back up a second, when did you decide to pursue biology, biochemistry to begin with? What kind of drove you in that direction in high school or whatever?
No, as a matter of fact, my brother had a lifelong interest in biology and actually went to medical school, as that was his prime thrust in life. I was really not, I did not view myself as a medical doctor. I viewed myself as perhaps more of an instructor, researcher, and really loved chemistry. I was one of those that got a chemistry set when I was 7 years old, and that really set the stage.
One of those Gilbert.
Yes, yes. So I was all set to do that. But as I went through graduate school, I saw the really a real major trend happening on the campus and in the literature of the application of chemistry and biology. I had no idea about recombinant technologies or any of the real foundations of it. I just had a sense that applying my knowledge in chemistry in a medical background made sense to me. So I actually went against my own sort of foundations and said, well, okay, I guess I can be a medical doctor, and I can show you the place on the UC Berkeley campus where from one step to another, I decided I'm going to medical school.
It just sort of coalesced and
it just coalesced. Yes.
That was a moment, wasn't it?
It was, I've had 3 epiphanies in my life, and that's one of the 3. And from that moment on, it was no looking back. And I was able to get into medical school, and found I really loved medicine. What really surprised me, it was not only the research, but I loved taking care of patients, and I still do.
That was no trivial. When you made that decision, when it came to you, that epiphany, your brother being pursuing the same thing, or already had, maybe older brother, you had pretty good knowledge of what investment of time and energy you were going to have to put in, not just academically, but then in residency and all the rest.
Right. Yeah. No, it was, like I said, I wouldn't allow myself to look backward. I would only look forward. And that was the way I was able to deal with it. My brother is 2 years younger than me, and he was 1 year ahead of me in medical school. So, he was always ribbing me as we went through medical school. So, but it was, medical school was a good experience. Learned a lot of biology and of course medicine, and was very happy to apply it during my residency.
Okay, so going forward then, you were doing research?
Right. Okay. There was an adjacent research center on the Presidio of San Francisco, and I was transferred there and worked for 2 years there. And toward the end of my time at the Letterman Army Institute of Research, I was talking with several people. I had, because I wanted to think about what my next step was. I did not feel like I should stay in the Army. I felt like I had a place in the civilian world. The only question was, what was that place? Well, a couple of different roads appeared to me. One was, I had been a volunteer physician while I was in the Army in American Samoa. and spent 3 months there. And of all things, my next door neighbor was the father of a budding rock star by the name of Huey Lewis. And his father, Hugh Craig, was a tremendous radiologist, and he was a volunteer physician in American Samoa. So here were two California boys that were next door to each other, a long way from home. And he thought I was a pretty good doc, and he introduced me to this very, very first-rate private practice in San Francisco. And I received an invitation to go into this private practice, which would have been very prestigious and, really kind of ordered my life. It seemed to be a pretty decent alternative. But then I was talking with one of my medical mentors, and we were talking about this new technology. It was absolutely wild. At the time, these two scientists at Oxford, Curler and Milstein, had developed a way of making what they called monoclonal antibodies. And we all know what antibodies are, because those are the things that are generated when you get a vaccine. But when you get a vaccine, you get many different kinds of antibodies. Well, they were able to figure out how to get a single antibody out of a single cell. And that may or may not sound important, but it opened up a whole new field of pharmaceutical drug development, because if you had one antibody, then you could make one drug. And that was very special. And what was important about these antibodies was the targeting ability, the ability of these antibodies to land on, say, a tumor cell and do something to that cell. So I decided, and this one is I don't understand myself. But I decided the only way I could do this was to start my own company. Now, you have to understand, I had no business background. The most I'd ever sold in my life, and this is true, are donuts as a Boy Scout. That was my sales experience. And I wasn't very good at it because I ate most of the donuts. But through contacts I had made, again, through the medical community that I was going in, I was able to get an audience with a number of potential investors. And what I realized at the time, and I think this was advice I recommend to everybody, and that is, if you have a good idea, someone's going to like it. And the key is, don't give it away to the first person that comes along. And I was smart enough to realize that if I talked enough, and my story got better and better, I would, perhaps land into a more favorable environment. And as I worked my way up the hill and got thrown out of a number of offices, I got pretty good at getting used to that. I finally landed on the doorstep of a venture capital firm called Hambrecht & Quist in San Francisco. And Hambrecht & Quist was really growing into this whole area of what was called biotechnology. That word didn't exist when I started knocking on doors. But they had just taken Genentech public, and that was an extremely successful public offering. So I landed, and there's nothing like luck, at a time when there was a very receptive audience. And we were able to, after a lot of going back and forth, land some venture capital money. And over the summer of 1981, I was able to raise, I think, $3 million, which back in those days was a lot of money. Yeah. And that got the company going.
So Pat, one thing, I didn't bring out earlier is, I worked in your organization for about 11 years running IT. And I actually have a science background, so I'm a chemist as well. But the one thing that I think people may not fully appreciate, you mentioned Genentech. They probably have heard of that name if they're not in the field, but there was only a handful of companies back then, if I'm not mistaken. So those were really early days in the application of this science to the medical field.
Yes, as a matter of fact, I believe, and I could be wrong on this, but I believe we were the first company to look at the therapeutic applications of monoclonal antibodies. If we weren't first, we were clearly in parallel with a very limited number of companies. There were a number of companies working on diagnostics of monoclonal antibodies, but we just decided to jump in feet first and try to see what we could do. And we were intrigued by, again, this protein's ability to target cells, and then we could deliver potential new therapies or potentially less toxic therapies. I think the background there is all of us, whether you're a physician or this just literally anybody, understands the terrible nature of chemotherapy and the nonspecific nature of chemotherapy. And the idea is to kill more cancer cells than normal cells. And it's a race to see, who wins. The idea with monoclonal antibodies, if we could target more of the toxic stuff to the cancer cell, then perhaps we could shift the therapeutic benefit. In many respects, we were way ahead of our time. Technologically, we were starting with relatively primitive molecular biology. And in those days, the only way to describe it was the Wild West. It's as much of a Wild West as I will ever know.
And that's true with any new technology. It's that curve, right? So you go up the curve as an early pioneer with a new paradigm, and you're working everything out on the fly.
And what you're simultaneously doing is developing new technologies and products. After all, our goal was to get something into the market and hopefully benefit patients. And I think one of the thoughts in my mind was, as a physician, how many patients can I treat in a lifetime? Well, I don't know, 10,000 or 5,000, I don't know. But if I got one drug into the market, I would reach out to more patients than I could ever dream of, in a private practice. So I was very motivated to try to weave our way through the changing technologies. And this won't mean much to your audience, but it'll give you a sense of the changing technologies. We started off with antibodies that were derived from mice. And then the next technology was what they called mouse-human antibodies that had part mouse, part human. Then it became more human-like, and then fully human. And all the time, this is happening over, say, a 15-year period, which was about how long that took. We were also trying to generate products. So it was exciting. Boredom was never one of our problems. And in the meantime, learning, what it means to go from, a venture capital company to sort of a pre-public offering company. And then in 1986, we went public, had a very successful public offering. And then now you're in the public domain where you're reporting regularly and looking back with great remembrance to the times when we were private and didn't have to do all this public accounting. And we were so all these parallel things going on at the same time was thrilling in many respects. We were writing new patents. We were taking these experimental drugs into clinic and so on. So it was really a very remarkable time.
Yeah, it's a very complex industry. for people who may not be in it, where and may not fully appreciate how expensive it is to develop a therapeutic, 10 years, $1 billion or $2, and you still don't know at the very end whether or not you're going to come up snake eyes, right? So,
right.
And often collaborations, I know XOMA had a number of different collaborations, and there are still, and XOMA has shifted in recent years to provide more of the technology and not be actively manufacturing and putting out clinical product. But all that work that was done, now what is this, going on 40 years ago where this started?
Just about.
Is still bearing fruit. It's still really moving forward.
I think monoclonal antibodies has become much more of a household word. I mean, at least it appears in the press almost on a daily basis, because people are trying to make antibodies to the COVID virus, and so the word is the technology is out there, but it took a long time. And bringing in and motivating young scientists to come on board and really try to pave new roads for possible clinical application. Very exciting times. It wasn't always happy, exciting. We had our clinical failures, as you might expect. We had our clinical successes. You're right about relationships. We had relationships with numerous major pharmaceutical companies and big ones, and as well, in sort of interesting manner, we had a very successful relationship with Genentech, and were able to get a drug approved in collaboration with Genentech. So there was this opportunity to also sort of peek behind the curtains of how these big companies were run, and even big biotech companies and how they were run, and that all built on my experience. And so, but as time moves on and a few decades go by, I felt like after 35 years I had put in my time and decided in 2016 to go ahead and retire from that. And that was about the time that the company decided to change the business model. So it was a sort of a win-win. I think I'd put in about what I thought I could put in, and it was time to move on. So that's what I've done.
Even be a little segue point there, but even before that point in time, one of the business units, if you will, one of the areas was in the area of biodefense that XOMA got involved in. And I know you personally took a real interest in that and helped lead that. So maybe you can talk about that, if you will, transition, another area that we highlighted.
So that was a transition that was very important to me at the time and continues to be important. In 1996, as I recall, we had a visit from the director of a unit in the Department of Defense called DARPA, the Defense Advanced Research Projects Agency. To be honest with you, I'd never heard of it. I mean, it seems funny these days. It's such a commonly known agency. This very forward-looking experimental unit that really crosses multiple scientific disciplines to enable new technologies and applications in defense of our country, and ultimately spin out into the civilian community in many ways. But I had this visitor, and he was going from company to company, trying to get a sense of how biotech companies worked. And he knocked on our door, and they said, and so they gave him to me, and I kind of said, okay, I'll talk to the guy. But we really hit it off. I mean, it was clear something in my mind and his mind were able to link up and say, we could do something together. And he recommended and nominated me for something called the Defense Sciences Research Council, which is a group of multidiscipline group of academics, mostly academics and a few commercial folks that get together and brainstorm for DARPA. And so I was able to join that Defense Sciences Research Council, and one of the first things that I became involved in was technologies that could be applicable to defending against what was called bioterrorism, more broadly biothreats, whether they are, as you put it, intentional or natural or accidental. Our role, if you think about it, those three categories really don't matter, because those three categories are more related to attribution than response. And so the goal here was to try to figure out how could you rapidly respond to, say, a pandemic, and what technologies could you bring to bear. Rapid drug development in the classical or traditional pharmaceutical world is an oxymoronic term. There is no such thing as rapid drug development. And there's a good reason for it. Drugs take a long time because they first have to build upon a long safety database, both in preclinical studies and clinical studies. And then you have to demonstrate efficacy in multiple clinical studies. Even studies that get to the last stage of clinical testing, so-called phase 3 studies, about 50% of the drugs even fail at that point, and that's after spending $1 billion or so. And what was interesting, the number you say $1 billion, I went back a few years ago and looked at the most successful biotech companies, and that $1 billion of raising $1 billion was a threshold that needed to be crossed in order to have sufficient momentum and investment to permit successful drugs. So what's great about America was that little companies that could start from absolutely nowhere could have an opportunity through the financial community to raise money, and based on serial success, ultimately become very large and established companies. And companies like Genentech and Amgen are great examples of that. So anyway, to get back to DARPA, I began working on ways of trying to modify or take advantage of technologies that might be applied, because the biggest challenge in biothreats, any biothreat, is not necessarily known ones. We know a bunch of them, like anthrax and smallpox, but it's the unknown. I mean, the knowns, most of the known threats have some sort of therapeutic response available to it. Certainly smallpox and anthrax do, for example. And there's been a long history of trying to develop therapies to known or suspected potential biothreats. But it was the one that I became interested in was the unknowns. What do you do when something comes that no one has ever seen before and you have no therapies? And if truly pharmaceutical drug development cannot be rapid, what can be done to make it rapid? And that became a parallel interest of mine now for more than 25 years. It is not a simple task, as you might imagine, but I started with DARPA, and I'm very proud of the fact I was able to contribute to a number of programs that DARPA funded ultimately that advanced the concept of accelerated manufacturing of pharmaceuticals, some of which are being applied today in the face of this terrible COVID epidemic. So I was as the interest in this grew, I was transferred, so to speak, from the from DARPA to another agency with the alluring name of the Defense Threat Reduction Agency. And I received a presidential nomination from President George W. Bush, and became chair of the Chem Bio Warfare Defense organization, and was an outside reviewer of a massive investment by the Department of Defense in that area, and was able to move that forward a good bit. And but then as presidential nominations are, you kind of go on when a president nominates you, and you go off when the next president comes off. No harm, no foul. It was time, so I was able to move off of that. But then I received the nomination from HHS Secretary Kathleen Sebelius to be a founding member of a new organization called the National Biodefense Research Council, and so I joined that and was able to contribute to that program on the civilian side, and of course over time I rotated off of that, and I've worked with a number of other federal agencies along the way to help move some of those programs forward. So that was, and the really cool thing about XOMA was they gave me the time to run this in parallel. It meant that I worked 7 days a week, but
still
that just didn't bother me. It was too exciting.
Well, that's an interesting thing. I mean, just to pluck that one thing out is when you are in a space that you have passion about, or where you feel a connection to your purpose, there isn't a loss of energy. And one of the signals where you might not be in the right area is you feel like you're pushing a rock up a hill all the time. Then it's a good time to step back and reflect. But as you've gone through that, and you went through the bioterror area, just seeing what progress was being made, was that a confirmation that you were in the right place? That was something you should be doing? Just wondering the moments associated with that lane, if you will.
You never know. But I had a feeling when I joined the Defense Sciences Research Council, and I was now amongst these great academics from Harvard and Caltech and MIT and a number of major institutions, and they all had disciplines that contributed, and I was able to bring in my contributions, because after all, I was bringing knowledge in the area of biotechnology that was brand new. And so I fit right in with that august body of scientists, and really learned a lot about the advantage of cross-discipline science, and what advantages you can take of that. And so it really became very exciting for me, and ultimately fed back to the company, because I was able to bring in a biodefense program that I think ultimately brought about $150 million of NIH money into the company for a generation of some particular class of monoclonal antibodies. So it was a win-win situation.
Yeah, I think just one last thought on that for the non-scientific types is that, people sometimes get confused about the annual flu vaccine, for example, and how that comes about, and how come I took a flu shot but I still got sick, right? It kind of maps over to my understanding of what you were developing at XOMA in that business unit, where you actually had to have a cocktail of multiple different, I think, serotypes, different antibodies to cover the spread of what might occur. Is that accurate?
That's accurate.
And so, essentially, I think the thrust was you developed the technology there, developed those serotypes, all the manufacturing support, everything to go along with it, in preparation for the potential stockpiling and having that available. I mean, this should be encouraging to people who listen in to know that most people don't know any of this goes on,
Oh yeah.
that there's all, because it's not a product like go to your doctor, get a prescription, or go and see something over the counter that you can fight something. All this stuff goes on in terms of readiness. And even though we've had this pandemic event, some of the technologies that XOMA developed and other companies really did make it possible for us to respond much quicker than we would have,
Yes.
previously.
Yes, the market, if you will, is the national stockpile, where a large reservoir of key medical and medically related items are stockpiled in regional centers across the country.
Just the national stockpile, is that true?
Yes.
Okay.
And the national stockpile was there before biothreats. It was there for any national emergency or regional emergency which would overwhelm the local medical community. But this was an important part of it, to be able to respond to these natural, intentional, or accidental threats. And so the company had, I think, a meaningful role in that. We were able to bring together a lot of technologies to make that work. And I know the team that has done that, and I am very proud of that accomplishment. And while we were also moving forward on the sort of, if you will, civilian, regular medical applications of the technology.
Well, if those two areas weren't enough, actually, I think preceding the biodefense focus that you had, you got involved with something else over in Palau. Do you want to talk about that?
Yeah. So in 1993, let's see, that would have been about 13 years after I started XOMA. I had hardly taken a vacation. One of the employees in the company, you might remember Chip Lambert, he convinced me that I ought to take up scuba diving. And scuba diving was interesting to me. I really enjoyed it. Although in diving off of Monterey, California, I just froze. I mean, it's cold water. I don't care. You can put all the dry suits on you want. It's cold water. And that wasn't exactly fun for me. I enjoyed it. But I had friends, Chip being one of them, who had gone scuba diving in the Republic of Palau. And I don't know if many of the viewers have heard of Palau, but certainly in 1993, I had no idea where it was. I just knew it was in the Pacific, and it was about the same distance above the equator as American Samoa was below the equator, so it meant a long ways away. And they were interested in historical scuba diving, and that rang a bell with me because I had both my father and mother were involved in World War II. My father was in the military. My mother grew up in Europe, and she actually during the war was a spy for the British government. So I kind of got two views of World War II as I grew up, and so this idea of historical scuba diving really grabbed me. And we had a specific target. And it turns out that in July of 1944, a young ensign by the name of George Herbert Walker Bush was flying his TBM Avenger over northern Palau and saw a Japanese armed trawler and dropped his bomb on it and sank it. And so our team was determined to see if we could find that trawler. It had never been located. And in 1993, it was starting to get close to the 50th anniversary of the end of World War II. And so there was a sort of a temporal reason to find it. And so we went off to Palau. And I'm glad I had been in American Samoa because I had some expectations, but Palau is an absolutely gorgeous country, beautiful water.
Well,
it is not diving in Monterey. It is diving.
I'm glad that it was a beautiful, but I'm intrigued by something. I'm glad it's a beautiful place. You couldn't just go and dive for pleasure. You have to have this goal in mind, right?
Yes, right.
Historical.
No, you're right. I don't think I would have. You're absolutely right. I would not have gone to Palau just to go diving in pretty water. That's right. I needed a goal, and I had one, and we found the ship, wow. And we were able to document it. And many years later, I was able to tell former President George Herbert Walker Bush my experience in finding it. But I came back, and my wife and I stayed in Palau a little bit longer after we found the crash, and or the wreck site, and we did something again that I honestly do not know why we did what we did, but we did it, and that is most normal people would have then proceeded to go scuba diving on some of the incredible coral reefs and other beautiful areas of Palau. And we hired a guy by the name of Lucky who and we said we want to look at wrecks from World War II, and we didn't just want to go to some of the shipwrecks. Had he found anything interesting? Well, it turned out Lucky had, and he took us into this backwater, and there was this little island all by itself, and lying next to the shoreline was a wing. It was a 65-foot wing. It had one engine on it, and it had an engine mount for a second engine, and it was just lying in the water, and it took my breath away. I immediately jumped into the water. It was lying in shallow, so I could wade to it. I quickly figured out that it was an American wing because on the engine it said General Electric, so General Electric was on there, and I was able to discern that it was probably a wing of a bomber called a B-24, because I had built a lot of model airplanes when I was a kid. And so if you have a 65-foot wing lying by itself, that can't be a good thing, right? I mean, obviously something bad happened here. And so I crawled all over the island and didn't find anything. I found a bunch of old Japanese sake bottles, but no rest of the plane. We drove the boat around the island, no plane. And I just couldn't put it down. I couldn't put this down any more than I could put down my interest in bio threats or starting the company. And so this was another epiphany. This was the moment. And again, just like walking on the campus and deciding I'm going to medical school, from one step to the other, I said, I don't know where this is going, but I'm gonna do something. And I through the most patient support possible from my very dear wife, in early 1994, I came back on my own, solo, had smarts enough to hire a local guide, not Lucky, but somebody else, and told him I want to find more. By then I realized that Palau actually had been the site of many military actions during World War II. There were 9 individual campaigns over Palau, and many planes were shot down, and I wanted to know more about this. And in particular, I became interested in what happened to the crews. I mean, again, you lose a wing 1,000 miles from your base, and you got nowhere to go. So I, started interviewing the elders who had recollections of seeing MIAs or seeing POWs and seeing planes crash in the water. And over a period of 6 or 7 years, I started to accumulate a lot of knowledge in my spare time about what happened there, and on an annual basis would go back and begin looking. About 2000, maybe 1999 or 2000. Well, let me back up. In 1996 or so, I was in a swamp, a mangrove swamp by myself, trying to locate an aircraft that I was told crashed in there, and I couldn't even get the locals to go in there with me. So I went in by myself, and let's just say I had a lot of problems. It was a tough swamp. I found the plane, took lots of photographs, and had a really hard time coming out. It was only later they told me that it was also the home of crocodiles, saltwater crocodiles, which are man-eaters, and which may be why they didn't want to go in there. I don't know. But the bottom line was, I said, I probably should not be doing this by myself. And I wonder if I could find people that would be interested in doing this with me. And that was the beginning of what we called the BentProp project. We named it that way out of respect because every crash site we ever found, we never found a straight propeller. They were always bent, and it was sort of symbolic of the tragedies that happened there. And we started to hold ceremonies, what we call flag ceremonies, where we would fold a flag and recognize the loss. And we had this idea that someday maybe we could give these flags to the families. And that led us to yet another Department of Defense agency, which I became very familiar with. And that it's now called the Defense WWII Accounting Agency, but it's gone through several evolutions. And back in those days, it was called the Central Identification Lab in Hawaii, or SILHI. And I made connections with them, and I said, I just want to let you know I'm doing this work and I'm identifying this. And their charter is a global effort to return Americans missing in action, either because of for combat losses or as POWs back to the United States. And we were definitely looking for planes that were likely to have remains on board. And so, and in 2004, after 10 years of looking, we were able to locate a bomber, another B-24 bomber that had the remains of eight Americans. And that was our first major MIA find. And we notified the agency, and they sent out Navy divers, and over a period of 3 years recovered all the remains. And in 2010, after going through all the DNA and other identification, there was a big ceremony at Arlington National Cemetery where they brought the families together. And the president of Palau actually came to the United States to be part of that ceremony. So it was an incredibly emotional time for our team to have that happen.
Extremely gratifying, I would imagine. Now, the other thing to interject for others listening in, and that is there's two things that folks could follow up on. Number one, a book came out of this experience. And number two, there was a 60 Minutes episode as well that you were interviewed on 60 Minutes to describe some of this work. Is that right?
Right. So the book is called Vanished. It was written by Will Hilton, who joined us. Took him 6 years. He actually went on field missions with us and followed us around. And the book came out about sort of the early years, if you will. It really does describe quite accurately the early efforts and team building and the gratification and reward we got of seeing families finally get unanswered questions. I think one of the things people don't realize is that unlike someone who's killed in action and returned home, where you can get closure, you know where that person is, you can go to the cemetery and pay your respects. An MIA family never, ever forgets. I've never met an MIA family yet that doesn't in some way acknowledge in their home a special spot for that missing person. And
they don't know whatever happened to them. I mean, the presumption is they didn't make it, but somewhere in the deep recesses of their thought process, they might be thinking, well, maybe they have another life over there somewhere.
I have on more than one occasion had families say, and they just hope that, number one, he was able to escape and be taken care of by the local natives, and number two, he must have had amnesia because that would be the reason he never came home. So I'm not going to call these fantasies. I would call those hope. Hope that there was some remote chance. And it's the difference between 99.999% sure that someone's lost versus 100%. And 100% means they were able to be returned home. All MIA families know is that they didn't, and it's possible that somehow through a miracle they might have survived. And we wanted to answer some of those questions. To take it a little bit further, we had run, I think, 16 or 17 formal missions in Palau, found a number of MIA sites, I think. The Defense Agency came out numerous times, found remains, not always, but some of the times found remains, and were able to, identify those remains for the families. But then in 2012, we met two scientists, Mark Moline, now at the University of Delaware, and Eric Terrell, who is at Scripps Institution of Oceanography, and they specialize in underwater robotics. They're oceanographers and have many interests, but one of their specialties is the application of underwater robotics, the autonomous underwater vehicles that have the capability to scan the ocean floor in a way that scuba divers can't. I mean, on a very good day in 60 feet of water, you can get a couple of 100 square meters if you're lucky, and really watch your dive tables and your dive computer, but you're not going to cover any kind of big area. And these autonomous underwater vehicles with sensitive cameras and side scanning sonar each one can cover multiple square kilometers a day, which is like a lifetime of scuba diving. And they can do it at depths that we can't even dream of. So they fell in love with what BentProp was doing. BentProp fell in love with their technologies, and these were really good guys. And we decided somehow, again, we didn't know how, we were going to join forces, and it just made sense. It was an application that they looked forward to of using their technology, and this was a way of extending our capabilities. And this was the genesis of Project Recover, which is the current organization. You can go to projectrecover.org and read all about it. And in 2014, I believe it was, that's when Anderson Cooper showed up in Palau, and he wanted to know what we were doing, and he did a 60-minute segment. And it was kind of fun working with Anderson. He actually dove on one of the crash sites with us, and That was an experience all by itself, having Anderson Cooper there, and that was one of the I'm told was one of the most watched segments on 60 Minutes up until that time. It was very popular.
Yeah, well, it's still out there. I think you can still view
you
it
can
on
still get it, I think,
or something. Yeah, it's pretty amazing. Do they cover the Scripps technology, the remote drone approach in that segment?
No, there's a little bit of it in there. There's actually a full-length documentary that has been filmed over the past 6 years that has not come out yet, but may be coming out later this year, that will take into account the technology side, the emotional side, the land experience, the underwater experience. It's a much more in-depth look, and that's about all I can say about it right now, because it's sort of not ready for prime time yet, but it's in the works. It's all done. The film is basically in the can. They're just trying to figure out when it will be coming out and how to distribute it and all kinds of stuff I don't understand. So, this went just like XOMA and just like, the Biodefense. This kind of started off with, I don't know where this is going, but this is something I can't put down. Each of these moments are very special in my life, because there was no sure thing about any of these. The BentProp Project Recover could have ended with, a nice recollection of an interesting vacation in a beautiful place, and that would have been it. And I just decided, no, there were too many unanswered questions, and I needed to know more.
So it wasn't that you didn't meet challenges along the way, and you were still running, part of the executive team at XOMA. You're still running a major company, pursuing a major passion of yours, actually two different passions, and then this is a third lane or branch. So all that was still going on. So there was something inside that was really drawing you forward, I would say.
Absolutely. And, it's not like I feel like there's some wind-up thing in me that I'm aware of. It's just when it happens, I'm alert to it. And it doesn't happen very often, thank goodness, because then I'd be, who knows. But, when it happens.
Just a little bit. So do you actually, as the years have gone on, do you actually pause and think, is this another one? I'm just wondering how you assess.
Yeah, that happens.
People are going to be, people have had transition thrust on them right now because of all the job loss. There's always people that I talk to in my coaching who are interested in, maybe this isn't the right career for me, or how can I advance and go further? And they're sort of exploring in general terms without something being thrust upon them. So understanding practically, or at least what your understanding is from your process, your journey, I think is very helpful.
Well, I think looking back on starting the company, XOMA, I sort of had the technical foundation. I'd be grossly underqualified these days because of the advances in molecular biology, but none of that existed. And it was a confluence of my recognition of the potential of the technology, and actually the sort of early days of that technology that made it work. And there was a certain excitement, the certain amount of, I don't know where this is going, but I think there's something to this that made me turn down essentially a very, promising career in the practice of medicine, at least in clinical practice, and go into something that was extremely high risk. The area of bio threats and dealing with how to respond to pandemics brought together again this paradox of you need new drugs, but you have to make them rapidly. And so, how's that going to work? How do you do that? I mean, this was, and I wouldn't expect people to understand that necessarily that the pharmaceutical industry is a conservative industry. It takes a long time to develop a drug, and to ask the field to compress something that's taking normally years and do it in months or weeks is just unprecedented. And yet today, that's exactly what's happening with COVID. A normal pathway to a vaccine is 10 years. And that involves hundreds of, animal studies, various kinds of clinical studies, all kinds of data gathering that because vaccines are generally given to healthy people. So it's not even regular drugs are given to sick people. Vaccines are now given to healthy people. So you really can't accept, things that you could accept in a therapeutic mode, you can't accept in the vaccine world. And so that's it. It takes a long time to develop a safe and efficacious vaccine. And yet, I think through some of the groundwork that many people, certainly I was just a small part of this whole experience of trying to think about how do you accelerate manufacturing in the face of a pandemic. I think what's happening now is we did learn something over the years, and we're trying now to expand upon it in ways that have never been done before. I think that what's coming up is, I don't know when the vaccine is going to come out, but I think there will be one or more vaccines, over the next few months. If fortune is on our side, it'll be before the end of the year. If not, it'll be sometime, certainly in the first half of next year. And, that's just, that's light
speed.
That's light speed.
That really is.
So yeah. we are, I think we're in a time where where people have to start thinking this way. And, if COVID is a pandemic that has been thrust upon us, there's, a lesson we need to learn from that. And that is, it's likely that whether the next one is COVID or something else, it's likely to happen again. And I think as a nation, we're going to be far more prepared. We will learn an enormous amount from this. And it's sort of gratifying to have played, a part in that. So I've felt very fortunate as an individual to pursue 3 completely different paths, none of which I was particularly, prepared for or trained for, but have been able to successfully contribute, maybe through enthusiasm and persistence. Persistence is a good word, by the way.
Yeah.
In moving these areas forward.
Pat, I really appreciate your time today. I mean, such a great story. And I think that, it was my pleasure to be able to be part of the XOMA team for the time I was there. And I saw you often.
You did.
And I do remember you going on your annual summer jaunts to Palau to pursue this other area. And I do remember the folks there going, what's he doing? The key message here is that there was a spark. There was some kind of confirmation deep inside of you. And I don't think anything was going to stop you from moving forward with it. And I think there's a lesson there, if nothing else, from some of these things discussed today. So again,
I will tell the viewers it was a real pleasure working with you, Joe. And you were beyond helpful to me to make sure that my ability to communicate with a computer was kept going, because without you, I would have been at a loss to keep up with the various IT technologies that we were seeing.
Well, it was a whole team there. And so I just wanted to wrap up, everyone, just by saying that please do check out projectrecover.org. to track the work that Pat is doing with the recovery and in that one lane or what one passion. Also, would like to ask if this is something that served you well, please subscribe to this podcast and post a review if you would. That would be very generous. And then also, additionally, if you're interested in additional resources that I might be able to provide to you beyond what I can post here today, then I will make those available on my website, www.titansoftransition.com, titansoftransition.com. And thanks again, Pat. I really appreciate your time today.
Okay. Thank you.